We're going to start our session 2, Research to Support Guidance Development for Topical Drug Products. Our 3 speakers are going to be Dr. Megan Kelchen, Senior Pharmacologist with the Office of Research and Standards within OGD. Then we'll have Dr. Eleftherios Sakalokou, Lead Pharmacologist from the Office of Research and Standards, and forgive me for not pronouncing it correctly. And then we'll have Dr. Tanaz Ramazanli. I'm trying to talk so fast, forgive me. Senior Pharmacologist with the Office of Research and Standards. So let us begin with welcoming our first speaker, Dr. Megan Kelchen.
Good morning, my name is Megan Kelchen, and I will be starting the session by providing an overview of the current trends and product-specific guidances for topical products applied to the skin. In my presentation today, I'll first describe how research contributes to the development and revision of product-specific guidances for topical products applied to the skin, and then identify some scenarios where it may be beneficial to get feedback from the agency during product development. So let's first talk about some bioequivalence approaches for topical products. The first is comparative clinical endpoint bioequivalence study. This is an in vivo study in which the test product is shown to be as efficacious as the RS, and both products are shown to be as or superior to a placebo control. The next is a characterization-based bioequivalence approach. This is a weight of evidence approach that compares the formulation, microstructure, and performance of the test and RS product. Next is a vasoconstrictor study. This is an in vivo study comparing the skin blanching effect of the test product versus the RS for corticosteroid products. And finally, a waiver of in vivo studies. And a waiver is typically recommended for products that are solution-based where bioavailability is considered to be self-evident. And for the purposes of the discussion today, I want to highlight that these bottom 3 approaches that are shown in the box here are considered to be efficient bioequivalence approaches. So shown here are some bubbles that include the 4 bioequivalence approaches I just discussed. And you can see here that in the overlapping section that many PSGs for topical products include multiple bioequivalence approaches. So for example, PSGs that recommend a comparative clinical endpoint study often include an efficient bioequivalence approach, such as a characterization-based approach or a waiver of in vivo bioequivalence studies. Now, when we think about how to prioritize PSG development or revision, it's important to note that we first prioritize PSGs for newly approved NDA products. And then we focus on prioritizing PSG revisions for products where efficient bioequivalence approaches are not yet available. So in the context of this slide, the highest priority for PSG revision at this time are PSGs where we only recommend a comparative clinical endpoint study. Now, it's important to note that if a PSG does not include a characterization, or if a PSG only includes a characterization-based bioequivalence approach, that doesn't necessarily mean that an alternative, more efficient approach would not be acceptable to the agency. It may just mean that we have not yet revised that PSG yet. And so we encourage you to discuss any proposed alternative approaches with the agency through a product development meeting request. Let's turn our attention now to characterization-based bioequivalence approaches. I've talked about the components of a characterization-based bioequivalence approach in detail at last year's SBIA presentation. But briefly, at this time in PSGs for topical products, we commonly recommend no significant difference in formulation, comparative Q3 characterization, and an IVRT study. And then depending on the complexity of the dosage form, we may also recommend an IVPT study or systemic PK study. And so focusing in on the no significant difference standard in a bit more detail, it's important to note that the no significant difference standard is a comparison of the test formulation compared to the RS. And it's based on the principles for assessing Q1, Q2 sameness, but it also considers certain differences that have been previously determined to be acceptable based on scientific evidence. And I've given a separate presentation that focuses only on the no significant difference if you want to learn more. So in PSGs for topical products, currently we recommend bioequivalence approaches for products that meet the criteria for Q3 sameness, which would be a characterization-based bioequivalence approach. And we also recommend bioequivalence approaches for products that meet the Q3 difference criteria, which would be a clinical endpoint study or a vasoconstrictor study. I want to mention that we are currently developing bioequivalence approaches for these Q3 similar products, and those efforts are currently underway. So OGD is constantly investing in research to streamline PSG recommendations over time across similar products. And I'll discuss a few examples of this in the next slides. But I want to mention that other examples of research will be presented at other meetings this fall, for example, at the AAPS meeting in October. So let's first start with the PSG for topical aqueous gels. This slide is a slide I presented at last year's SBIA presentation that shows the components of a characterization-based bioequivalence approach in PSGs for topical aqueous gels. And so for the purposes of today's discussion, we'll use globule size distribution as a measure for the complexity of the dosage form. And you can see that for the majority of these products, when we do not recommend globule size distribution, we also do not recommend an IVPT or PK study. And the deviation from this trend is Dapsone topical gel. In September of 2023, we included additional recommendations such as IVPT and PK for these products based on our understanding of the site and mechanism of action of this drug product. However, over the last year or so, we've developed a more comprehensive understanding of the structure of the drug product, as well as the impact of this structure on the performance. And Dr. Sacalozzi will talk about how we use modeling and simulation to do this in the next presentation. But for now, I'll just note that based on this research, we were able to revise the PSG in February of 2024, removing the recommendation for IVPT and a PK study from the characterization-based bioequivalence approach. And so now here on the side, you can see the recommendations for topical aqueous gels for a characterization-based approach as of today. And so the lesson from this example is that OGD is constantly working to streamline our recommendations so that we have similar recommendations for similar products. Now let's turn to the PSG for clascoterone topical cream. This was first recommended in November of 2021 with a comparative clinical endpoint study. And we worked with our colleagues in the Office of Pharmaceutical Quality to develop an IVPT method that may be used as part of a characterization-based bioequivalence approach. And as part of this method development, we optimized various parameters such as the apparatus, the receptor solution composition and volume, the sampling method and volume of sample, as well as the study duration. And so based on this research that we did in the lab, we were able to revise the PSG in August of 2023 to add a characterization-based bioequivalence approach that includes a recommendation for IVPT. I want to note that the OGD maintains a list of PSGs that we plan on publishing within the next 12 months. And this list includes PSGs that we plan to develop as new PSGs, as well as PSGs that we plan to revise. The key take-home point for this first part of my presentation is that OGD is constantly working to streamline recommendations across similar products within PSGs based on cutting-edge research. Now let's switch to when you might want to seek feedback from the agency and how to do that. So one aspect of your product development program where you might want feedback is on the formulation. For example, you may want feedback on the proposed concentrations of an inactive ingredient. The agency's inactive ingredient database, or IID, can be used to help justify levels of inactive ingredients in a proposed test formulation. And it's important to consider the context of use when selecting concentrations of inactive ingredients. For example, the route of administration, context of use, or duration of use, and patient population. But I'll note that while the route of administration is listed in the IID, the duration of use and the patient population are not. And so we strongly encourage you to discuss your proposed concentrations or your proposed formulation with the agency early in product development. And this can be important regardless of the bioequivalence approach that you intend to use. For example, before conducting a comparative clinical endpoint study, we encourage you to receive feedback on your proposed concentrations before going into the in vivo study. Another aspect where you might want feedback on your formulation is a no significant difference assessment. As I mentioned, I've previously given a presentation that focuses on this evaluation a couple of years ago. And in session 6 tomorrow, we'll have a presentation that discusses what should all be included in your formulation assessment submission. So in the interest of time today, I will just note that it's important to include all relevant information related to your formulation in your submission. I want to turn now briefly to TDS products. The PSGs for these products are very consistent in that they all recommend an in vivo bioequivalence study, an in vivo adhesion study, and an in vivo skin irritation and sensitization study. And in April of 2023, we revised this the guidance for industry on irritation and sensitization potential. And in that revised guidance for industry, we note that in some circumstances, an in vivo sensitization evaluation of a TDS product may be unnecessary if adequate justification is provided. And so if you want feedback on a proposed alternative study design compared to what's in the product-specific guidance for these products, we encourage you to submit communication to the agency through a controlled correspondence or a product development meeting request. You might also want feedback on your proposed bioequivalence approaches and your proposed study design. So if you're developing a product that is intended to be Q3 the same as the RS, we encourage you to submit a product development meeting request if no PSG is available, or if a characterization-based approach is not yet available in the PSG of the product you intend to develop. You may also want to send in specific questions about that you may have about the design or conduct of specific studies, such as the IVRT or IVPT studies. And Dr. Ghosh will be discussing these studies in more detail in session 6 tomorrow afternoon. Now, let's say you submitted a formulation for assessment as part of a controlled correspondence, and you get feedback that your proposed formulation may not qualify for a characterization-based bioequivalence approach under the Q3 sameness standard. But you would like to move forward with that formulation anyways. Then you may have a Q3 similar product, and we encourage you to submit questions about the studies that may be used to support this alternative approach in a product development meeting request. Finally, we encourage you to submit communication through a controlled correspondence or a product development meeting request if you intend to do a comparative clinical endpoint study, if the PSG is not available, or if you have specific questions about the design or conduct of the study. And finally, you may have noticed that in recent years, we've been adding this language to PSGs that indicate when an RLD product is a drug device combination product, what the device constituent parts are, and what information should be submitted in your ANDA for assessment. However, I want to note that for topical products, we have not yet included this language into all of the relevant PSGs. And so if you have questions about whether an RLD product for a topical product is a drug device combination product, we encourage you to send in a controlled correspondence to get clarification. And drug device combination products will be discussed in more detail in session 4 this afternoon. But for topical products, a tube or a pump that does not alter the dosage form or does not meter the dose are considered to be container closure systems rather than devices. Alternatively, a pump that changes the dosage form, for example, from a solution to a foam, or that meters the dose, a TDS product that's meant to adhere to the skin, or a pledgette or swab is considered to be a drug device combination product and would require comparative analyses of the user interface of the test product and RLD. So the take-home point for this final section of my presentation is the pre-ANDA program can be utilized to get feedback from the agency during both early and late stage development, regardless of the bioequivalence approach you intend to use. So let's move to some challenge questions. Which of the following are considered to be an inefficient bioequivalence approach for topical products and is therefore prioritized for PSG revision? A comparative clinical endpoint study. B, a characterization-based bioequivalence approach. C, a VC study. Or D, a waiver of in vivo studies. The answer is a comparative clinical endpoint study. Which scenario would benefit from a pre-ANDA interaction with the agency? A, the PSG recommends a comparative clinical endpoint study, but you would like to use a characterization-based bioequivalence approach. B, it is not clear from the PSG if the product is a drug device combination product. Or D, or C, you would like feedback on the proposed levels of inactive ingredients prior to conducting a comparative clinical endpoint study. Or D, all of the above. The answer is D, all of the above. So in summary, the PSG recommendations provided by OGD are streamlined recommendations across similar products based on cutting edge research. And engaging with the agency through the pre-ANDA program to gain feedback through product development can be beneficial regardless of the bioequivalence approach you intend to use. I'd like to acknowledge all of my colleagues at the FDA, and here is a list of resources that may be used for reference. So with that, I'd like to thank you for your attention. I look forward to addressing any questions that you may have during the panel session, and I'll invite Dr. Sacalozzi for the next presentation. Thank you.
Hello, everyone. Thank you, Megan, for the introduction. I would like to now take this opportunity to discuss a case study where we use modeling and simulation to enhance our understanding of structure-performance relationships for Dapsone topical gels. The learning objectives for today, I will discuss in silico methodologies for topical products that are applied on the skin. I will explain the data requirements and scientific considerations when building and validating dermal physiologically based pharmacokinetic, PBPK in short, models, in this case for Dapsone topical gels. And we will recognize how in silico models can be used to support regulatory decision making. The outline of the talk, Dr. Kelchen did an excellent job describing what is the rationale, the thinking behind developing product-specific guidances, what are the considerations. I will just highlight some relevant ones, relevant to the specific example I'm discussing. And then I will give a little bit of information on what are in silico methodologies for topical products. And I will spend most of the time talking about the case study, a dermal PBPK model that was developed for Dapsone topical gel, 7.5 and 5%. What were the considerations that went into the development, validation, and the application of the model? So PSGs for topical products contain harmonized recommendations that are in alignment with relevant general guidances, such as the Q3 characterization guidance, the IVRT studies for topical drug products guidance, and the IVPT studies for topical drug products guidance, all submitted for ANDAs. When the PHGs contain recommendations within a characterization based by equivalence BE approach, then these BE recommendations are product specific and they're based on an understanding of the microstructure and performance of the drug product. I believe that Dr. Kelchen made that clear. However, BE recommendations in the product-specific guidances may also include in vivo BE studies with PK endpoints or comparative clinical endpoint studies. So this talk will focus on a revision, PSG revision, that took place in February 2024. And this PSG revision refers to two products, the Dapsone topical gel 7.5% and the Dapsone topical gel 5%. Prior to February 2024, the PSG contained two options for this product. So for test products that met the no significant difference criterion, what was recommended was an in vitro characterization approach that required Q3 sameness and a comparative IVRT and IVPT passing studies. And this approach was supplemented by an in vivo BE study with PK endpoints. The option 2 was reserved for test products that did not meet the no significant difference criterion. After February 2024, this guidance was revised, and option 1 does not include anymore an IVPT study or an in vivo BE study with PK endpoints. The rest of the in vitro characterization studies under option 1 were maintained. This PSG revision, as Dr. Kelchen pointed out, was the result of our improved understanding on the product structure, microstructure, but it was also supported by modeling and simulation. This approach, the modeling and simulation approach, elucidated the relationship between drug product quality attributes, which are the Q3 characteristics that are included in our PSGs, and the in vivo performance of the product. And these models that I will go into a lot more detail next establish the link between systemic and local dapsone exposure. So very, very quickly, the in silico methodologies that I will be discussing today are essentially mathematical statistical models that describe how the amount of dapsone in this case changes with time based on an administered dose. To generate this output, these models take into account information on the in vitro performance of a product. This could be, for instance, an IVPT study in the case of the Dapsone gel. Information on the physiology, the skin, what are the physiological parameters that impact permeation through the skin and distribution throughout the entire human body. And also, these methodologies account for drug product characterization information. So a particle size distribution would be an example of this type of information. Now, for this case, we developed two models for these Dapsone gels, different strengths, 7.5 and 5%. The objectives of these models were to understand the factors that are governing the permeation of dapsone through the skin, identify which drug product quality attributes that are specific to the dapsone topical gels may impact local and systemic bioavailability. And these models were finally applied to perform virtual bioequivalence assessments that evaluate the impact of formulation differences on BE outcomes. These virtual bioequivalence assessments were performed at the level of the plasma, so systemic exposure, following the application of the gels, and also within the context of an IVPT study, an in silico IVPT study. In order to develop these models, we leverage data on the in vivo permeation, the in vivo absorption of dapsone gels, and that is described through the model, the schematic on the top here. But also, at the same time, we leveraged information from IVPT studies by developing an in silico IVPT model that really captured all the experimental conditions that are relevant to an IVPT study. These models work together to develop and validate each other and result in reliable, realistic predictions of dapsone permeation through the skin and absorption into the systemic circulation. So this is the general workflow that we followed. It is very similar between a topical gel, the 7.5 and the 5% strength. So we started with developing a systemic disposition model for dapsone. We leveraged literature data to do that. And then we started focusing more on the skin absorption component of that model and developed the dermal PBPK model for the 7.5% gel. We used data from approved ANDAs to optimize certain components of the model. And that optimized model then was validated against additional independent data sets that were extracted from other approved, additional approved ANDAs. Once this model was validated, we applied it towards virtual bioequivalence assessment for all ANDAs that were considered for development and validation. And in addition to the plasma BE assessment, we also were able to perform bioequivalence assessment in the stratum corneum and the dermis for this product. What was also unique in this specific case is that we were able to validate the model using IVPT data. And in my next slide, I will go into a lot more detail on this. The workflow for the development and validation of the Dapsone topical gel 5% model is very similar. We started with the same systemic disposition model. And now we switched to ANDAs, approved ANDAs for the Dapsone topical gel 5%. We saw that the model for the 7.5% topical gel performed really well. So all the data from the approved ANDAs for the 5% was considered external validation. We did not need to optimize the model at all for the 5% strength. And similarly to what we did earlier with a higher strength, in this case, we applied the model for VBE assessments for the ANDAs, approved ANDAs for the 5% strength in the stratum corneum and dermis, so locally and systemically. Now, we just discussed the workflow for the 7.5% gel, and I said that we used IVPT data to further validate this model. So in parallel to this specific in vivo model, we developed an in silico IVPT model, which described the IVPT study conducted for the Dapsone topical gel 7.5% within an approved ANDA. We optimized certain parameters of the model. I will describe those in my next slide. And we used that partially validated IVPT model, in silico IVPT model, to assess by equivalence between the RS and the test product in the skin, the stratum corneum, and also the receptor media. So this is a schematic that will help you understand how these models are built. They're essentially mathematical equations that describe how the API is partitioning, is being released from the formulation, which is the gray compartment here, into the stratum corneum and the rest of the skin layers. In order to describe mathematically this process, we can account for Q3 aspects of the product, such as formulation pH, particle size distribution, apparent viscosity, and solubility. So these are direct inputs into the model, and there's information that is collected within the scope of the Q3 characterization of the product. Nothing really is required in addition to those Q3 studies. We do account for formulation composition when we are building these models. As I mentioned, the Q3 aspects, the drug product quality information is included into the model. And for validation, we used in this case in vivo systemic PK data and IVPT study data. We made certain assumptions regarding the evaporation and drying rate of the gels, but this can also be characterized experimentally. And in the case of the in vivo application, we did not assume any rubbing, although this was reported in the clinical study protocols. So we utilized these models for VBE assessments. We performed 10 virtual BE trials where we matched essentially the in vivo BE studies that were conducted in support of the approved ANDAs we worked with. And we assessed by equivalence virtually for Cmax and AUC in the stratum corneum, the dermis, and the plasma. In terms of the IVPT studies, we again matched the conditions of the IVPT study in terms of donor demographics, replicates, and dosing conditions, and the experimental conditions. And we assessed B for maximum amount and AUC in the stratum corneum for Dapsone and cumulative amount and Jmax of Dapsone in the receptor solution. This slide summarizes the performance of the model, where we see that overall the blue lines that are the model predictions overlay relatively well with the observed data, which is the black dots. Now, when it comes to IVPT data, we developed an IVPT model, as I mentioned earlier, and in this case, what we see, the model predictions are the red and blue lines, and the observed data are the squares. What was interesting here, and I would like to note, is that what we saw is that when we include variability in the applied formulation on the receptor, on the donor side, we were able to describe the variability that we see in these type of studies very well. So that's an interesting point to consider in the future. Similarly, in the case of the 5% gel, we're summarizing here the performance of the model, predictions and observed data overlay relatively well. So these models were applied to assess bioequivalence between the RS and the test product in the stratum corneum, the dermis, and the plasma across all the ANDAs that we utilize for model development and validation. And we saw that when we have a test product that meets the no significant difference criterion, Q3 sameness between those two products leads to BE locally and systemically. And certain Q3 attributes, in this case particle size distribution and apparent viscosity, impact BE outcomes and should be closely controlled. Same were the outcomes in the case of the virtual IVPT studies, where we looked at Jmax and the cumulative amount in the receptor solution and also amount of Dapsone in the stratum corneum. All of these ANDAs were considered, were found to be bioequivalent, and similarly to the in vivo study, this modeling and simulation approach showed that Q3 sameness between the reference and the test leads to BE locally and the receptor solution. So in conclusion, this modeling approach improved our understanding of the structure-performance relationship of Dapsone gels. Apparent viscosity and particle size were shown to be important Q3 quality attributes. And these developed models can serve as tools supporting a risk-based assessment. The compelling evidence from this PBPK modeling approach aligns well and reinforces the BE recommendations in the aforementioned draft PSGs for other topical gels, as Dr. Kelchen pointed out. So first challenge question: which Q3 attributes were identified as impacting bioavailability based on the developed dermal PBPK model? Formulation pH, rheological properties, particle size distribution, and droplet size distribution. And the answer is B and C. Challenge question number 2. Enhanced understanding of the structure-performance relationship of Dapsone gels gained through the PBPK modeling research led to a successful product development meeting for an applicant, an ANDA approval, or a PSG revision. And the answer is a PSG revision. Thank you very much for your attention. I would like to thank everyone, my colleagues at DTP1 and DQMM, for their support on this project. And we can move on to the next talk that will be delivered by Dr. Tanaz Ramizanli on the approaches for evaluation of formulation differences on performance of topical products. Thank you.
Good morning, everyone. This is the last presentation in session 2, and I'm going to talk about the approaches we have been working on to evaluate the impact of formulation differences on performance of topical products. The objectives for my presentation are first to describe consideration for formulation development of a test product that does not meet the no significant difference criterion. Second, to evaluate the impact of compositional differences or Q2 differences on bioavailability of topical gels. And the last is to evaluate the impact of Q2 changes on sensory perception of topical gels. So you heard from Dr. Kelchen's presentation earlier in this session about the bioequivalence approaches that are currently recommended in our published PSGs for topical products. In this diagram, you can see the potential strategies for BE that was developed or are under development under GDUFA Regulatory Science Program. And they have grouped them based on the requirement in the for the formulation. So when a test product, for example, meets the no significant difference criteria and it is pre-qualified for a characterization-based approach, and this approach is currently recommended in many of our PSGs for topical products. But when there are differences in a formulation of a test product compared to reference standard, then there are other approaches that we have been working on to develop for this category of test formulation. So that there are approaches based on cutaneous pharmacokinetic studies, such as ones using dermal microdialysis or dermal open flow microperfusions that are in more advanced stages of development. And there are others that are in earlier stages of development, and we are still investigating them, such as cutaneous PK studies using imaging or spectroscopy-based tools, or the ones that is the focus of the today's presentation is to evaluate the impact of formulation differences on thermodynamic activity and sensorial perception of topical products to be able to expand the characterization-based approach to some of these test formulations that may have compositional differences compared to the reference standard. So we first needed to understand the function of excipients and their impact on thermodynamic activity of the drug in topical formulations. And we started with single-phase gels, which are the least complex form among the semi-solid topical dosage forms. And in the figure, you can see different types of excipients that may exist in a single-phase gel. We have solubility modifiers such as propylene glycol, polyethylene glycol, or glycerin. We have gelling agent, preservative, pH modifier, solvent, etc. And among these different types of excipients, we identified that probably the Q2 changes in solubility modifiers may have the highest impact on thermodynamic activity. So we started focusing on the solubility modifiers in our research. And in order to predict thermodynamic activity changes, we first needed to define fractional solubility, or alpha, which can be calculated from concentration of solute divided by saturation solubility of the drug or solute in the solvent. For example, if we have PEG water formulations, by increasing the amount of PEG in the formulation, since it is a solubility modifier, the solubility of the drug of interest most likely would increase. For example, here in this example, the saturation solubility of this drug has increased by increasing PEG concentration. But if we have a constant drug concentration, according to this equation, by increasing the denominator, the fractional solubility would decrease. So by increasing PEG concentration, the saturation solubility increases, but at 7.5 milligram per ml concentration, we have the lowest alpha with the 40% PEG formulation. And fractional solubility is often predictive of thermodynamic activity of the drug in the formulation and how likely the drug molecules would leave the formulation and partition into the skin. So one of the earlier studies that our collaborators did was to make series of propylene glycol water formulations. These were like 2-ingredient formulations, and the saturation solubility of diclofenac sodium, a lipophilic drug, was measured in series of this formulation with variant amount of PG from 0% to 100%, and as expected, by increasing in PG amount, the saturation solubility increased. Then several of these formulations were selected in the range of 10% PG to 50% PG, and they conducted an infinite dose IVPT study. So as also explained in the previous slide, when we have a constant drug concentration, for example, in this case, 2% diclofenac sodium, by increasing the amount of solubility modifier, we expected to see a decrease in the thermodynamic activity. And the IVPT results also show that with 50% PG, we got the lowest flux, steady state flux, and with 10% PG, we had the highest steady state flux. I would like to know that since we had infinite dose in this IVPT studies, the metamorphosis of the dosage form was negligible, but when the topical product is actually applied on the skin by human or by patients. The finite dose is used, which undergo metamorphosis and drying, and following solvent evaporation, there may be changes in drug solubility at the application site, or even change in microstructure and Q3 properties, for example, change in rheological properties of the formulation as the formulation dries, and all of them may lead to change in drug permeation and bioavailability. So while we can measure fractional solubility at time 0, we didn't know how this fractional solubility or thermodynamic activity would change across the product application on the skin. So what we did that our collaborators, they monitor the drug solubility or saturation solubility at the application site by measuring drug concentration in a donor compartment. So they basically had the Franz diffusion cell set up similar to how the IVPT is set up, and they used a semi-finite dose on the skin in the donor compartment, but instead of sampling the receptor compartment, they sampled the donor compartment and measured drug concentration in the donor compartment at different time points. So using this methodology, we would be able to have fractional solubility profile across the duration of application here, for example, for 6 hours. And here shows the results we got with HEC gel formulations, which included PEG 200 as a solubility modifier at different concentration, 1%, 10%, and 20%. And here the drug model was diclofenac sodium and with a fixed concentration. So as is shown here, with different amount of PEG, we had different fractional solubility, and during the course of 6-hour application, the fractional solubility with 1% PEG increased, and for the other two it kept constant. But what is important is we saw a well-separated profiles from the 1% and 10% and 20%, which suggests that the thermodynamic activity of the drug in these 3 formulations are different, and subsequently the IVPT results also showed that the permeation rate was significantly different from for the diclofenac from the 1% PEG formulation compared to 10% and also 20%. The similar experiments were conducted with HEC gels that contained metronidazole. So this is a hydrophilic molecule with different pKa and different physicochemical properties. And here, interestingly, with the variation in amount of PEG from like 1%, 10%, 20%, did not result in significant differences in fractional solubility at time 0 and also during the course of 6 hours. So subsequently, the permeation profile was also very similar for these for metronidazole from these 3 formulations. So we were able to establish a nice correlation between the drug bioavailability and the thermodynamic activity. Next, we wanted to focus on smaller Q2 changes and their impacts on bioavailability. So here shows examples of gel formulations where PG, propylene glycol, was used as a solubility modifier for both metronidazole formulations and diclofenac sodium formulations. And we first evaluated the saturation solubility of the drug in different amount of PG. And we, for identifying the reference product, we selected portion of this curve where it is relatively steep and it's within the range of PG amount used in approved topical products. So for metronidazole, the reference formulation contained 30% PG, but for diclofenac, the reference formulation contained 40% PG. And then from there, we calculated the PG amount in plus minus 10% difference and plus minus 25% difference in PG, and make those formulations with metronidazole and diclofenac. So the fractional solubility measurements showed that with plus minus 10% variation in PG, the thermodynamic activity is not changing, and subsequently the permeation rate is not changing compared to the reference formulation, but with plus minus 25% difference, the fractional solubility has changed, and the flux profiles also are distinguished from the one for the reference formulation. With diclofenac sodium formulation, while we see some separation of for the plus minus 10% formulation than compared to the reference, the IVPT result did not show significant difference in bioavailability of diclofenac for the plus minus 10% variant formulations and the reference formulation, but the profiles for plus minus 25% variant formulation were different compared to the reference formulation. Next, we wanted to understand whether the Q2 changes in topical products would have other impacts beyond changing the bioavailability. For example, would differences in Q1, Q2, Q3 of topical products result in differences on how the product would perceived by human subject and potentially change in therapeutic equivalence. So for that, we needed to establish a correlation between Q2, Q3, and sensory perception of topical products. And also we needed to understand whether the characterization of Q3 properties, such as rheological behavior of topical products, predict the sensorial differences that may be perceived by subjects. So for that, our collaborators did develop objective tools to measure some relevant Q3 attributes and also conducted in vivo sensory panel tests of those formulations to be able to correlate these Q3 with those sensory attributes measured. So here we also started with single-phase gels, and we identified relevant sensory attributes of those gels. In the left image or figure shows the list of sensory attributes that we evaluated for topical gels, like spreadability, smoothness, shine, cooling sensation, etc. And we identified which of them potentially could have more impact on therapeutic equivalent. So for this study design, after identifying the sensory attributes, we needed to find for each sensory attribute what would be the relevant Q3 attributes that we can measure in vitro, and how we can measure that using what kind of instrumental technique, and also potentially what formulation variables can more impact the changes in these Q3 attributes and the sensory attributes. For example, for something like a cooling sensation, potentially a drying rate study was assumed to be a relevant test to inform about the cooling potential of a formulation, and probably the change in amount of solvents or co-solvent may have an impact on changing these sensory attributes. And for something like spreadability, we assume that rheological behavior of the gels, like measurement of zero shear viscosity, yield stress, etc., with a rheometer would inform the change in the spreadability, and potentially the amount of gelling agents would have the highest impact in changing these Q3 attributes and this sensory attribute. So here are the results for the Q3 test. Some of them are shown here, and also the in vivo panel study for these three attributes that are shown here. For several carbopol 980 gel formulations that were made with different composition of a gelling agent and alcohol. And in this figure, the data is normalized for each unit, for each attribute, in the range of 0% dark blue up to 100% yellow. Formulation 2 here was the reference formulation. And for example, in formulation 3 and 1, we had differences in the amount of carbopol. Formulation 3 had like double the amount, and formulation 1 had half of the amount used in formulation 2. And these changes in the gelling agent resulted in significant change in the zero shear viscosity for these two formulations. But only in formulation 3, we observed a significant difference in the perception of stickiness and spreadability compared to formulation 2. And in formulation 4 that we had alcohol as opposed to formulation 2, the slipperiness of this formulation was perceived significantly differently. The similar studies was done with hydroxyethyl cellulose gel with different compositions of PG and propylene glycol and alcohol. Here, formulation 3 was the reference formulation, and the variation in the amount of alcohol in formulation 1 to 4 resulted in significant change in zero shear viscosity. And in formulation 2, we had significant difference in perception of slipperiness. In formulation 4, where both the amount of propylene glycol and ethanol was different compared to reference formulation, the stickiness of the product was also felt differently. In conclusion, this preliminary results for this gel suggest that when we have Q2 changes, we may or may not see significant differences in sensory attributes. But when we have significant differences in Q3 attributes, we likely would see some, or we may see, or it's more probable that we see differences in sensory perception of those products. So with that, I summarize that FDA is investigating alternative scientifically valid methods, including in vitro approaches to support assessment of BE for topical products that have compositional differences compared to reference standard. The current research data suggests that when there are differences in thermodynamic activity, such differences may result in bioavailability, as shown as evaluated by IVPT. The Q3 properties assessed instrumentally in vitro may be valuable in understanding most of the sensorial differences for topical gels in vivo. And the current data suggests that large differences in Q3 attributes, such as for like neurological, tribological, or textural properties, would likely perceivable to human subject. And finally, research is underway to further evaluate the impact of Q2 differences on bioavailability and product perception of topical gel formulations. So here are my challenge questions. The first question is, which statement is not correct? Q2 differences would always result in changes in performance of topical products. B, it may be feasible to assess fractional solubility of the drug in conjunction with IVPT to assess impact of Q2 changes on performance of the product. C, significant Q2 differences may result in changes in performance of the topical product. And D, metamorphosis of the formulation following topical application may change the microstructure of the product. So the correct answer is A, because Q2 differences would not always result in changes in performance of the topical product. Then second question is, which of the following Q3 attributes is more likely to correlate with spreadability and stickiness of topical gels was based on the data I presented? A, friction of coefficient, B, zero shear viscosity, C, drying rate, D, all of the above. And the correct answer is zero shear viscosity. So with that, I would like to acknowledge and thank my colleagues and supervisors at US FDA and our research collaborators. And thank you very much for your attention, and we'll be happy to answer to your questions during the panel session. Thank you very much.
Thank you very much. We call our speakers back to the panel table, and we would like to welcome two new panelists, Dr. Hiran Patel, Senior Staff Fellow from the Office of Bioequivalence at OGD, and also Dr. Pahala Simamora, Division Director of the Office of Product Quality Assessment in the Office of Pharmaceutical Quality. So we'll do the same thing like we did last time. Any in-house questions, please come up to the microphones. We have such a lovely panel of experts here. They're just waiting to answer your questions. Brave, sir. Yes. I hope you're going to the microphone. Okay, yeah.
Yeah.
All right, question at mic 1, please.
Marie Ducharme, Learning Confirm in University of Montreal. I have a question for Dr. Tsakalozu regarding, first of all, it's a comment, and then the other one is a question. The comment is, you said that the model established a link between systemic and topical concentration. Isn't that an assumption of the model?
It characterized the relationship, right?
Well, it's an assumption of the model. It's built in.
Yeah, it's part of the structure of the model. Yes.
Yeah. Okay. So I think the sentence is a little bit misleading. I mean, just a comment.
Sure. Thank you.
The question is, what is your validation criteria? You know, you showed, validation with data and all of that. What is it?
Yeah, that's an excellent question. So you're asking this question because the performance of the model was not always ideal, right? So the challenge that we typically see in PBPK modeling, and it was very, very obvious with this type of work that I presented here was that there was a lot of variability in the in vivo PK data. The study conditions varied between the ANDAs that were utilized for model validation in terms of the population, the application sites, the doses that were applied, and the conditions. So rubbing, for instance, is a big consideration that unfortunately currently we don't take into consideration into the model. And this is a condition that may impact skin permeation to a great extent. The other challenge specifically for this work is that the application site was not the same across all of these studies. So that introduced variability in our model predictions. The criteria that we used in terms of the internal validation is a 20% difference between the predicted and the observed. We want to be as close as possible when it comes to the internal validation, but for data sets that are used for external validation, there are parameters that come into play in terms of the population variability that we cannot always account into our model, and that leads us to the model performance that you saw here.
Yeah, but what was the criteria? So it was like minus to plus threefold, or what
Yes.
was it?
It was twofold.
Twofold? For the mean?
Yes.
The mean being twofold? Okay.
Yes.
Thank you.
Thank you, Dr. I'm going to get this. Dr. Sakalaka Luzu. I'm just saying thanks. Okay. Any in-house questions? Any? Okay, let's go online, please.
Okay. We have actually another question for Dr. Eleftheria Sakalozu. How are in silico models being validated for a given drug product and indication? If the same model is being used for another product, what variation will have to be considered?
Thank you. That's another good question. So the models that mechanistic models, such as the one that I discussed today, are drug product specific. So they're built utilizing data that are specific to the product of interest, and then they are validated either with IVPT, microdialysis, open flow microperfusion, or in vivo PK data that are also collected after following application of that specific product. So in other words, in the example that I described here today, for every ANDA, two models were developed, one for the reference standard and one for the test product. The validation is performed across all the available data sets. Obviously, we're looking for data sets that are good quality, that are collected under controlled studies. And against the previous question, obviously there is a variability around those data sets. I hope that I've answered this question. Hopefully I didn't miss anything. Thank you.
Thank you. Next online question, please. We'll just continue with the online unless somebody comes up to the mic.
Next question is directed to Dr. Pahala Simamura. Could you please elaborate on the usefulness of in vitro characterization and in silico approaches to support SUPAC changes?
Thank you. That's such a good question. As we know, SUPAC guidance has been in place since in the 90s, so it's been a while. It's been evolving so far, as Dr. Megan Kelchen presented, all the evolvement to date. A lot more we learn about drug product, whether it's simple or complex, and so on and so forth, right? So, and also, as the previous section mentioned about looking at the totality of the data as well. The point is, I think with a strong product development, strong product understanding and process understanding, I think it would make it easy when post-approval we want to make changing to certain thing. Is there like a specific things to do? Maybe not. Maybe it's a combination of things to look at the totality of the information. I don't know if I hope I answered the question. I know it's quite generic, but compared to the what we have under, which is currently is the standard for post-approval change. But I think now we have identified, a lot more parameters that are more meaningful for to identify, to mitigate the risk of changing certain things post-approval. Thank you.
Thank you. Next question, please.
Next question is for Dr. Megan Kelchin. Do I need to get feedback on a proposed concentration in my test product if the level is below what is listed in the inactive ingredient database, or IID?
Thank you for the question, and thanks for the opportunity to clarify this. So the external IID is a great starting point when developing or trying to figure out which concentrations you want to use in your proposed test formulation. Because as I mentioned in my presentation, the IID includes the route of administration for the drug product. So the idea being that your proposed concentration should be below the level that's included in the inactive ingredient database for that route of administration. But as I mentioned, the context of use, so for example, the duration of use or the patient population that product is used on or used in is not listed in the IID. And so what we don't want to do is use a product that is for an acute indication to justify the levels of a product that, or a concentration that's to be used in a product for a chronic indication. And so while it's not necessarily required, we strongly encourage you to come in with your proposed concentrations and get feedback from the agency. And again, this can be helpful even if you want to do an in vivo study, because we don't want you to do a comparative clinical endpoint study just to find out your proposed concentration is too high compared to what we have in-house. Thank you.
Thank you, Dr. Kelchin. Next question, please.
Next question is for Dr. Hiren Patel. Could you please highlight the criteria for recommending IVPT in the in vitro characterization BE approaches? Also, how can an applicant interact with the agency for feedback and recommendations?
Sure. Thank you for the question. It's an excellent question. As we heard, Dr. Kelchin talked about how the characterization-based BE approach laid out. It based on the complexity of the drug product. For example, and it is, as we heard in the earlier session too, that it's a weight of evidence-based approach. So it is how we systematically mitigate the risk of failure mode. For example, if the drug product is a single-phase formulation, then if the formulation is meet the no significant difference criteria, which is based on the Q1, Q2, have similar Q3. And if you conduct IVRT study, we believe that it systematically mitigates the risk of failure mode. There are certain exemptions. For example, if it has a complex inactive ingredient like white petrolatum, then we may recommend IVPT study, even though it's a single phase formulation. However, if the formulation is a multiphasic, we typically recommend IVPT study in addition to the Q1, Q2, Q3. However, there may be some exception on multiphasic formulation. For example, if there is no clinical relevance, we may waive the IVPT study. So I hope that answered the question about in which instances we recommend IVPT study. The second part of the question is if you seek agency's feedback either on the utility of the characterization-based approach, I would encourage you to submit product development or control correspondence. Thank you.
Thank you, Dr. Patel. We do have an in-house question, please. Go ahead.
Yeah, I have a practical question. Let's say if you use topical drug applied to skin and for a certain time, and you continue measure plasma concentration on those things. So there's a timing difference beyond how when you stop use drug. When you do modeling to the validation and how long time you need to take after the drug finish.
That's a question for me.
Yeah.
Okay. Yes. So the model predictions are considered validated for as long as there is observed data to track, to compare those simulations against the observed data. So if you have an observed PK profile that is 12 hours, but your model is predicting for 48, clearly the part of the model predictions that are not validated against, they're not compared against observed data, are not as credible as the ones that are compared to actual observed data, right? There are, when we are assessing model validation, when we're assessing model performance, we're looking for the overall shape of the curve we're interested in, right? So Cmax, AUC, Tmax, and then additional characteristics that may be relevant to the product. Multiple peaks, maybe.
Can I follow up?
Please.
So in certain cases, some drugs, they have a faster metabolism rate. And even when they continue release, you might see drug in skin tissue, but the plasma concentration can go down instantly to 0. And in that situation, you don't see too much drug concentration in plasma, but you still know drug is releasing and you see in tissue. And a validated modeling is going to be what timing cutoff. And even we should use plasma to do the modeling at all. Just wanted your thought about that. I don't know what's right or wrong, just your thought.
Yeah, the case that you're describing is definitely challenging. It would be very useful in this case to follow a more mechanistic approach, modeling approach, I mean, where there's additional information that goes into the model that can justify, that can support further the model predictions. Then it will also be helpful to show that this same model or similar model or modeling suite is also behaving well with other products. So the uncertainty that is present in your own case is in some way mitigated by accumulating knowledge in other cases. Additional areas like matrices where the model can be validated would also be useful. Additional sources like multiple studies will also be validated, will also be useful. Best practices when we are developing a model, overall best practices present in our guidances should be followed to kind of mitigate, to increase our confidence and mitigate that uncertainty, that risk that we have with the lack of the data. It's a very general question.
Thank you very much for that response. Online, please.
We have an online question for Dr. Tanaz Ramzanli. How does comparing metamorphosis and thermodynamics translate into a patient's or prescriber's concern about similar look and feel of a topically applied dermatologic drug product?
So as I shown in the presentation, one of actually our research project focuses on that and to see how the differences in Q composition and Q3 properties can change the sensorial perception of topical products. So our early results or like preliminary results suggest that we can correlate these sensory attributes with in vitro Q3 tests. And our in vitro Q3 tests are usually more like strict to differentiate compared to how the differences will be significantly felt by patients. So, so eventually, I think if we have differences in the composition of the formulation, and the formulation is not gonna go through the clinical endpoint study where it's tested in patients, there might be alternative ways that we can mitigate this failure modes, and we are working to develop the alternative studies for that.
Thank you. Next question, please.
Next question is for Dr. Sakalozu. Could you comment on whether the US FDA develops models or methods for assessment of in silico studies, or prefer that the manufacturer do so?
Thank you for the question. It really depends on what is the need that a product presents, an application presents. If an applicant identifies the need for their product to be supported by in silico approach, we welcome this. We will call it alternative B approach, and we will we'll provide the necessary feedback. In cases where we feel that there is a risk with the product, in cases where we want to inform our regulatory policies, such as the guidances, we would conduct internally modeling and simulation. So both.
Thank you. I think we have time for one more online.
This is a general question for the panel, or for anyone on the panel. PSG revisions are a big headache for industry. Therefore, why don't you publish a set of correct studies to begin with, rather than revising them one at a time based on approved ANDAs?
So I'll start, and others can chime in. So the recommendations we have for in each PSG is product specific and is recommended based on our evaluation of the data for that product and understanding the complexity of the product. So we definitely cannot and would not recommend a general studies for series of different products, because each guidance or product specific guidance is specific to that particular drug product.
Thank you. Do you want to squeeze in one more online?
Can I actually add something?
Sure.
I'll note that when we publish PSGs for newly approved drug products, sometimes we want to do some additional research before we publish, say, a characterization-based approach. And so we might publish a comparative clinical endpoint study as part of the PSG to get the guidance out there, to give industry an opportunity to have a PSG available. But there might be some internal research that we're doing, such as like the clascoterone cream that I discussed in my presentation. Thank you.
Okay, I think we'll call it, we're just on 11:45. So thank you to our panel.